All right, last video. So we left off at 5' and 3'. We'll look at this again in class together, but it's going to be really important that you understand how to identify the 5' and 3' sides. You should be able to look at a model like this of a DNA molecule, look at the oxygens, think of them as arrows pointing to that 5' side. The reason it is that because when DNA replicates, it matters what side we're looking at. If we're looking at the 3' side or if we're looking at the 5' side, because they're going to do something a little different. So the last thing we're going to look at is we're going to review a little bit of our DNA and RNA structure. So here's kind of a brief overview, and then we'll go to this one. So big things we should know, and this should be a review for you guys, is that DNA is double-stranded. It has two sides. RNA is going to be single-stranded. The other thing that's different about them is we have DNA and RNA. The NA in those acronyms refer to nucleic acid. It classifies them as what type of organic macromolecule they are. But that first letter, deoxyribose for DNA and ribose for RNA, is a designator for what type of sugar we find in our nucleotide. Now you'll notice the highlighted difference right here, which should be a reminder for us. The reason it's called deoxyribose is because you notice in the blue circle, you're missing an oxygen. Whereas on the ribose sugar, you have that oxygen right there on your 2' or your second carbon. 2' carbon over here, missing an oxygen. The last thing, and this is going to be really helpful for you guys when you are designating and looking at the differences between DNA and RNA, because like I said, it is your nucleotide sequence, those A's, T's, G's, and C's, or in the case of RNA, those A, U's, G's, and C's, that are going to be communicated when we talk about DNA sequences. So you have to be able to know that when you see thymine, you're looking at a DNA sequence. And when you see uracil, you're looking at an RNA sequence, because RNA doesn't have thymine and DNA doesn't have uracil. All right, last check-in. Which is stronger, the bond between nucleotides or a phosphodiester bond? All right, so you should be able to look back at your notes. It's the phosphodiester bond, which forms between your phosphate group and your 3' sugar, that is going to be stronger because it's a covalent bond. Remember, hydrogen bonds, they're just weak associations. Does the following nucleotide sequence come from a strand of DNA or RNA? All right, so this is what I was talking about as far as identifying based on nucleotide sequencing here. So we have A, T, T, C, G, C, C, A. So you should be able to look at this. G's and C's don't tell you anything. A's don't tell you anything actually either, because we find adenine, cytosine, and guanine in both our RNA and our DNA. So you got to look for it. If you see a T, then you know it's DNA. So I see them, I know it's DNA. Tough one. If a double-stranded segment of DNA contains 26% guanine, what percentage of the strand is cytosine? All right, so the answer to this is 26. What we have to remember here is that guanine and cytosine pair up with one another, which means you're always going to have exactly as much guanine as you do cytosine. Because if it's double-stranded, then every G has to pair up with a C. Now, this is only true for double-stranded DNA. If something's single-stranded, then we don't have that pairing. And I would never ask you a question like that because that would be very tricky and mean. But you should realize that it's the pairing in the double-stranded DNA that gives you information about this. All right, the last thing I wanted to share with you guys, because this is helpful for kind of remembering the DNA organization levels, is you can kind of think of the different ways that DNA is organized by making an analogy to a book. So if you think of the genome, so all of the genetic information in an organism, all of it, think of that as the entire book. But then that book is going to be organized into chapters, so segments of a larger story. And those are like the chromosomes. And then within those chapters, we have paragraphs. We're going to have sort of short actions, little things that go on. And those are like your genes. Those are the small recipes that make up our chromosomes, that make up the genome. But each of those paragraphs is made up of letters. So much like the English alphabet is made up of 26 letters, our genetic alphabet is made up of 4 nucleotides, A, T, G, and C. How we vary those letters is going to determine the code. It's going to determine what word, or in the case of genetics, what gene we're trying to express. All right, so make sure you filled in your guided notes. Email me if you have any questions. The next thing we're going to look at is we'll look at DNA replication. So we'll get into the actual enzymes, the process itself.